So I want to bring your attention to some drugs that affect the neural control of skeletal muscles. And you don't need to memorize the names of these drugs, but what I want to point out is really the effects. Because I think that really helps us to understand better what, how the nervous system works. So the first drug, the botulinum toxin, inhibits the release of acetylcholine. So you can imagine if acetylcholine can't be released by the presynaptic nerve, then the acetylcholine won't be able to produce an EPSP or the IPSP, right? Depending if it's going to bind to the nicotinic acetylcholine receptor or the muscarinic acetylcholine receptor. Another drug, which I mentioned before, the Curare, prevents the interaction of acetylcholine with its nicotinic receptor proteins, right? So if it can't interact with the receptor, then we won't have the EPSP or IPSP produced. Another mechanism for drugs is to bind the acetylcholine receptor proteins and prevent acetylcholine from binding, right? That would be a competitor of the acetylcholine for the receptor. That's one way to prevent the signaling. Another way is to block the voltage-gated sodium channels, right? That would be for the action potential. So even if there was these EPSPs, if the voltage-gated sodium channels are blocked, there will be no action potential. Another problem could be caused by nerve gas, for example, which inhibits acetylcholinesterase in the postsynaptic membrane. That would lead to either too much EPSP or IPSP, and that's really problematic. And then another one prevents the IPSPs in spinal cord that inhibit contraction of antagonistic muscles. For your muscle to contract, you have some muscles have to pull and other muscles have to relax. And basically, if the muscle that needs to relax can't relax, then you can't have movement. So next, I'd like to move on to the amino acid neurotransmitters. So glutamate is an excitatory neurotransmitter. It's the major excitatory neurotransmitter in the brain. It produces EPSPs in about 80% of the synapses in the cerebral cortex. And astrocytes are really important in taking excess glutamate from the synaptic cleft, because if there's too much glutamate there, that can cause toxicity, right? That's problematic. That's why the acetylcholinesterase is so important. And in this case, the astrocytes are so important to remove the excess glutamate. So the glutamate receptors are also ion channels, and these play a key role in memory storage. And some of these are excitatory and some are inhibitory. The major inhibitory neurotransmitter in the brain is GABA, and GABA receptors contain a chloride channel. So let's look at the GABA receptor. So the channel is closed until the receptors bind to GABA. Okay, there's just like the nicotinic acetylcholine receptor, there's two binding sites. So if GABA binds the two binding sites, what will happen is the GABA receptor will open, and this happens to be a chloride channel, and then chloride ions will be moving from the outside the cell to the inside the cell following the concentration gradient. So in all these channels that are being controlled in the EPSPs and IPSPs, the ion is always traveling with its concentration gradient. So I mentioned that GABA is an inhibitory neurotransmitter, meaning it produces the IPSP, and that is because as the chloride ions come in, that is further going to make the membrane potential more negative. It's going to be lower, right? And that makes the action potential much less likely. So monoamines are another class of neurotransmitters. And these are neurotransmitters that are derived from amino acids. So for example, we have the catecholamines that are derived from tyrosine. This includes the important neurotransmitters dopamine, norepinephrine, and epinephrine. Indolamines are derived from tryptophan, and the one I'll be talking about today is called serotonin. So how are these monoamine neurotransmitters acting in the synapse, and how does the body control to make sure that there's not too much excess monoamine neurotransmitters in a synaptic cleft? So like the other neurotransmitters that we've talked about, the monoamine neurotransmitters are packaged in vesicles that need to fuse with the plasma membrane to be leaving by exocytosis, right? And just to remind you, there has to be an action potential. The action potential, the depolarization, right, due to the sodium, will lead to the voltage-gated calcium channels to open. Calcium will be following its concentration gradient, being carried from the outside of the cell to the inside of the cell. This will then complex with some proteins that are needed for these vesicles to exit the cell via exocytosis. So the neurotransmitter enters the synaptic cleft. They will interact with receptors in the postsynaptic cell. Right after the receptor does its job, right, they can do it over and over, but there's a mechanism to prevent that from happening. And for the monoamine neurotransmitters, the mechanism is to have the neurotransmitters be uptake by the presynaptic cell. After they enter the presynaptic cell, they can be inactivated by a monoamine oxidase, and that can then basically deactivate the monoamine neurotransmitter, and then it can be recycled. So again, this is really an important way. So we've studied so far acetylcholinesterase to prevent having too much acetylcholine. We talked about astrocytes preventing there being too much glutamate, and now we're seeing the reuptake and then monoamine oxidases that help to prevent there being too much of the monoamine neurotransmitters in a synaptic cleft. So let's take a look at the action of norepinephrine inside the postsynaptic cell. So here we have norepinephrine binding to a receptor. Its receptor in the postsynaptic cell. We see the G proteins, right? And to remind you that the G proteins form a trimer when the alpha subunit is bound to GDP. When the alpha subunit binds GTP, they separate. And in this case, the alpha subunit bound to GTP will be traveling to another enzyme that's a transmembrane protein called adenylate cyclase. And the alpha subunit bound to GTP will then activate adenylate cyclase. Adenylate cyclase will convert ATP to cyclic AMP. Cyclic AMP has the ability to convert an inactive protein kinase to an active protein kinase. A protein kinase can phosphorylate proteins inside the cell. So this protein kinase is going to phosphorylate this ion channel and then open it. And basically, this will then allow for ions to move in or out with their concentration gradient. So the next neurotransmitter I want to talk about is dopamine. And dopamine is a really important neurotransmitter. And one disease that we've all heard of that involves a problem with dopamine is Parkinson's disease. So Parkinson's disease is caused by the degeneration of motor control neurons that are producing dopamine in the brain, in the central nervous system. And basically, patients who have Parkinson's disease don't have enough dopamine. right? Because the parts of the brain that produce dopamine are dying. They're not able to produce high dopamine levels. So what patients are treated with, they're treated with a combination of L-DOPA and the monoamine oxidase inhibitors. So basically, L-DOPA can help to increase the amount of dopamine. It's a precursor for dopamine, and basically that can help these nerve cells produce more dopamine. And then they also have the inhibitors of the monoamine oxidases that break down dopamine in the presynaptic nerve cell. So basically, all these two mechanisms together work to increase the amount of dopamine found in the synapses. The other thing that dopamine does, in addition to controlling motor control, it also affects the emotional reward system which is localized to a different part of the brain. So dopamine plays, and it has an association with addiction, such as nicotine, alcohol, and other drugs. And if you have too much dopamine, someone can develop schizophrenia. So the interesting thing is that neurologists discovered that if you give a schizophrenic too much medication, that would basically reduce and lower dopamine levels. If dopamine levels get too low, those patients can develop Parkinson's-like disease symptoms. And then, critically, if a Parkinson's patient has too much of the drugs that are increasing dopamine levels, and the dopamine levels get to be too high, those patients can develop schizophrenic-like symptom. Yeah, so basically it's a very interesting combination of diseases related to dopamine. So basically, too much dopamine can lead to schizophrenia. Too little dopamine can lead to Parkinson's disease. And the amazing thing is that medicines for each of those different diseases can cause symptoms of the other disease. So it's a really interesting story and showing how important dopamine is both for the emotional reward system in the brain and also the motor control region of the brain. Another neurotransmitter to highlight is serotonin. And serotonin is implicated in mood, behavior, appetite, cerebral circulation. And the drug LSD and other hallucinogenic drugs are maybe agonists. And basically, serotonin levels have been also associated with, linked to depression. So if someone doesn't have enough serotonin, they can be depressed. And in fact, some common drugs for treating depression, including Prozac, Paxil, and Zoloft, these are all specific reuptake inhibitors of serotonin. And the idea is if the serotonin in the synaptic cleft cannot be reuptake by the presynaptic nerve cell, then those serotonin in the synapse can continue to bind their receptors in the postsynaptic cell. So what are some other types of neurotransmitters in the body? So we also have neuropeptides, and so neuropeptide neurotransmitters include CCK, which is involved in feeling full after a meal, substance P that mediates pain, neuropeptide Y that makes you want to keep eating, and opioids that can reduce pain and also lead to euphoria. So neuropeptides are released just like the other neurotransmitters we discussed. They're in vesicles and they can be released, and sometimes they're even released, just released from these vesicles, right? And it's going back to cell biology, right? Proteins that are destined for secretion will end up in these vesicles prior to secretion. And so these can be triggered in different conditions, and there's a lot of synaptic plasticity, which means the body can make a lot of adaptations to release different neurotransmitters under different situations.